Hello, my name is Will Spooner. I'm the CEO of Zeta Genomics, a company founded to commercialize the outputs of the OpenCGA project. And I'm delighted to be back at BOSC, albeit virtually, to present OpenCGA, the genome-optimized data store. Now, OpenCGA is open source software, and it has, in my opinion, the potential to transform the way that we manage and analyze genomic information. The context that I'm talking about here is the rise of precision and genomic medicine fueling the interest in the field of genomics. The Global Alliance for Genomics and Health, for instance, is predicting that there will be in excess of 100 million whole genomes sequenced within the next few years. There are more and more data sets are growing to very, very large sizes, joining the Genomics England 100,000 genomes program in their scale. And one of the significant challenges for the field is around the use of POSIX file systems, which are simply not appropriate for the management of data sets of that scale. The solution, of course, is to introduce the big data stack to genomics, and that is exactly what OpenCGA does. So if we think about this from a genomic workflow perspective, we have the current situation where we're writing our genome data files, our VCF files, into generic file storage, and that creates a number of challenges. First of all, the VCF files are effectively static text documents, and they're pretty much immutable once they've been written. A second issue is accessibility. The files are only really accessible by bioinformaticians who can understand the contents of the files, and the bioinformaticians also need access to the local file system. And then there's scalability. Any project over a couple of thousand whole genomes has to be split over a large number of VCF files. What that really does is makes the downstream analysis pipelines pretty linear, pretty inflexible, and what the pipelines generate themselves is yet more static text files. What we believe is that the generic file storage can be replaced pretty much wholesale by a genome-optimized data store, and this transforms the situation. So the new workflow is that you stream your genomic data into your data store, and it is normalized, deduplicated at ingestion. It is then linked and annotated within the data store, and it is transactional within the data store. Once in the data store, it becomes easily accessible over the internet using web tools, and the technologies that underlie the optimized data store are inherently ultra-scale technologies, and that allows a single repository to be deployed that is capable of holding pretty much unlimited volumes of genomic information in a single repository. Looking downstream of the data store, what used to be in the old world your analysis pipelines now becomes a real-time query against the database, which returns enriched and annotated information, and it's performed by the analysts. And so the interpretation, it becomes iterative, it becomes more collaborative, and that leads to improved decisions, improved insight, and much greater opportunities for data reuse. So OpenCGA is an optimized genome-optimized data store that uses a big data stack, and it's been developed over a 5-year collaboration between the University of Cambridge and Genomics England under the OpenCGA umbrella. So what is OpenCGA? Well, you can think of it as a platform that provides convenient access to a range of stakeholders to an integrated data set that contains the genotype data linked to the sample and clinical metadata, and linked to the reference annotation data, and the stakeholders access through an access layer, a set of data services that provide for programmatic access, web applications that provide point-and-click access, and also an authorization and authentication layer provides security to make sure that everything is kept secure. Okay, so what sort of things can OpenCGA can you load into OpenCGA? Well, the first thing is the output of your secondary bioinformatics analysis, your genotype data. And it can also integrate with lab information management systems and ingest sample metadata and then clinical and phenotypic metadata from, for example, electronic health record systems. These are all loaded and linked within the OpenCGA data store, and then they are annotated against information in a knowledge base. OpenCGA works with the cell-based knowledge base, which is also a product of the OpenCB project. So once everything is loaded and annotated within the system, it's then available for expert analysis. Expert analysis can be used for generating research insight. That research insight then eventually makes its way back into the knowledge bases, where it becomes available for further annotating the information in the data store. Other application area is clinical decision support, so use of the information in clinical interpretations. The results of those interpretations can then be loaded straight back into directly into the data store, and they provide further annotation of the information that's held. OpenCGA is a client-server architecture. So on the client side, you have your analysts on their workstations, and they can access and interact with the data in OpenCGA through the OpenCGA web application called IVA. There are client libraries for Python and for R, which allows programmers to code access to OpenCGA within their application code. And there's also an OpenCGA command line interface for convenient access direct from the terminal. All of the clients communicate with OpenCGA through a REST web interface, and the web interface is an interface onto the OpenCGA middleware, and the middleware then orchestrates the queries and requests to the data layer and also out to the cell-based knowledge base. So looking in a little bit more detail at the server side of OpenCGA, so we have the middleware, which is the web applications, the query planning, running asynchronous jobs, and the middleware then communicates with the data layer using over a service-oriented architecture. And there are a number of different data stores that we use that are used by OpenCGA, depending on the nature of the information that's being represented. So the genetic variants, the sample genotypes, and the variant annotation are very well represented in a wide column data store, and the software that is used is Apache HBase. The clinical and sample metadata are stored in a MongoDB document store, and then the wide column store and the document store are kind of glued together, if you like, using Solr search indexes. These provide query optimization, and they also enable combined genotype and phenotype queries against the integrated data. They're also from the catalog, as part of the metadata, are references to the source data files, the BAMs and CRAMs, and this enables the things like alignments to be extracted from the front end as well. So what does it look like in a real applications? Thinking about the 100,000 Genomes project, so the infrastructure that has been stood up for Genomes England consists of a cluster of about 30 machines, Kubernetes cluster, a Hadoop cluster, a couple of big VMs for Mongo, a couple of VMs for Solr, and so that's sort of the size of infrastructure that is needed for one of these very large genomics projects. Now I want to move on and show a bit of a demo what it looks like to query hundreds of millions of variants in real time via a web interface, and also showing you how the system is very well suited to querying the data at all scales, from many tens of thousands of whole genomes, or alternatively just down to a single interpretation of a single sample of a single clinical case. So this is IVA, the web user interface for OpenCGA, and the dataset that we're going to start with is Nomad Genomes. This is a summary level dataset that consists of about 70,000 whole genome sequences. And the first question is, how many unique variants are there in Nomad? And the answer is just over 700 million unique variants. So how do we drill down into that to find the number that satisfy a particular query? Now, the query that we're going to use is the pathogenic stop-gained variants in the ACMG incidental findings genes. So first thing we're going to do is we're going to set a filter for those variants that are annotated as pathogenic in ClinVar. The second thing we're going to do is we're going to set filter by the 59 genes on the ACMG panel. And the third filter that we're going to set is consequence type. We're only interested in stop gain. So we can go through our list of consequence types, stop gained, and so there we have our 3 filters set. And then we click search, and of those 700 million variants, and this is a real-time query against the OpenCGA web services, we find that there are 226 that satisfy our criteria. Now, Nomad is a summary level data set, so we're going to generate a few summary statistics using IVA's aggregation function, and we're going to aggregate by gene. So we're saying which of 226 variants, which genes are they found in, and so we find that 37 of them are found in BRCA2, and at the other extreme, there's only one found in some of these other genes at the other end of the distribution. Something we can do with IVA is we can save filters. So I'm going to save this filter. I'm going to call it BOSC. And I'm now going to take this filter and I'm going to apply it to a different data set. I'm going to apply it to the 1,000 genomes data set. We do have sample level information for this particular data set. There are 72 million unique variants in the 1,000 genomes, and we can pull up our filter that we've just saved, and so this is going to tell us how many pathogenic stop gained variants there are in the ACMG genes, and again, a real-time query, there's 13 of those. So I'm going to drill down, rather than try a summary aggregation, I'm going to drill down into information for a particular variant. I've selected this one here, which is in the stop gain of the SDHB gene. Having a look at the clinical annotation, what clinical annotation do we have about this variant? And we find that there's a number of different studies from ClinVar that suggest that this gene is associated with hereditary cancer predisposing syndrome, and also that it is monoallelic, so you only need one copy of the allele to express the phenotype. How common is this variant? We can have a look in our sample stats, and we find that there's just a single sample in this collection that is heterozygous for this particular variant. We could have a look at the beacon function to see which other data sources in the beacon network have this variant. But what I'm going to show you now is how we can drill down even further to the sample level using using IVA and the data in OpenCGA. So we've now moved into our the IVA case interpretation tool. I'm going to jump straight to the sample variant browser. I'm going to go back to filters. I'm going to pick up the pre-cam filter again. Right, BOSC. There's the BOSC filter. Click search, real-time query, and we find that there is just one, and it's the same one as we were looking at earlier, SDHB stop gained. And if I hover over the sample genotypes, we can find a display of the information that was extracted from the original VCF that was loaded into the system. So OpenCGA is completely non-lossy with regards to the VCF information that was loaded into it. In fact, you can think of OpenCGA to some extent as a VCF database. Okay, I'll stop the demo there and move back to the main presentation. So this is my last slide, and the purpose of this is to reiterate that OpenCGA is fantastic and open source software, and it's freely available from GitHub. It is developed and maintained by over 40 contributors worldwide. It is used by several very high-profile genomics initiatives. Genomics England, for instance, use OpenCGA in both their clinical pipeline and also in their trusted research environment, and also that the community, the OpenCGA community, is growing, and we'd be absolutely delighted for more people to come and really discover what it is like to run bioinformatics using a big data stack. So please do go over to GitHub and see what it's all about. Acknowledgements. Nacho Medina, who is the lead developer of OpenCB, he is founder of Zeta Genomics, and he's head of the University of Cambridge Computational Biology Laboratory, and he's really the driving force behind OpenCB and OpenCGA. At Zeta we have Jacobo, Pedro and Laura. At Genomics England, Antonio, Julie and Javier, and at the University of Cambridge there's Daniel and Herkin. So thank you very much for listening.